All right, so respiratory pigments. So, what are these respiratory proteins? They're essential for increasing the carrying capacity of oxygen in the blood, and they do this by binding to oxygen. Because once oxygen is bound to these pigments, it's no longer in solution. So the solubility does not limit the amount of content of oxygen in the blood. So the main thing to note is that these binding proteins for oxygen have metallic ions as part of their structure. Now, there are multiple different respiratory pigments distributed across different animals, and the features are a little bit different, but they all kind of have the same idea. So, I think everyone knows the big one, hemoglobin. It's the most widely distributed pigment, are present in 8 phyla. So, most vertebrates, some annelids, some mollusks, some small crustaceans, etc., nematodes. It's very, very widespread. Um, and this contains a porphyrin prosthetic group with an iron inserted, E, which is a heme. A porphyrin with an iron inserted is a heme. Each heme binds one O2 molecule, right? And they are found in corpuscles normally, extracellular in some vertebrates, but normally in most it is extracellular. In corpuscles, and it's red when oxygenated and blue when deoxygenated. So here we have a little bit of a better look at things. This is a hemoglobin. It's a tetramer. So we have 2 alpha and 2 beta subunits, and we have these heme groups. So you can see the beta subunits here, alpha here. You have these heme groups, which is basically this porphyrin group or this porphyrin ring with this iron in the middle. And this porphyrin group is basically just a modified structure composed of 4 pyrrole groups. So this is the structure we're looking at when we're talking about hemoglobin. Now, just a quick side note of some other pigments you might find. Hemerythrin. This is in like brachiopods, some worms and polychaetes. It's non-porphyrin, so there's no heme group, but there still is an iron. There's 2 iron atoms per O2 binding site, which is going to be different from the each heme binding 1 O2 molecule. Here we have 2 iron atoms per O2 binding site, non-porphyrin group. The protein is most commonly exists in an octamer, or 8, but dimeric, trimeric, and tetrameric forms are found in some animals, and it's colorless when deoxygenated and reddish-violet when oxygenated. And this is always in some form of corpuscle. We have hemocyanin, found in many species of invertebrates, but in only 2 phyla, most mollusks, including some bivalves, arthropods. There's no heme, porphyrin, or iron. It's copper-containing. And each subunit binds 1 oxygen molecule per 2 copper. It's hexameric in structure, and it's always in plasma. Colorless when deoxygenated and blue-gray when oxygenated, as in cyan. You see the cyan. So it's blue-gray when oxygenated. And then we have we'll get there. Chlorocruorin. Jesus Christ. This is found in polychaetes only. Iron-containing porphyrin structure, but different from the normal heme group. So it does contain a porphyrin structure with iron, but it's different from the normal heme. And it's always in the extracellular plasma. It's light red when oxygenated, greenish when deoxygenated. And these coloring is due to the unusual porphyrin structure. So, there is a diversity of the oxygen binding curves for blood, right? So, various patterns in oxygen saturation curves are explained by different properties and modifiers of the respective respiratory pigments. So, basically, the chemical structure, the chemistry of these pigments is going to affect the curve at which oxygen is binding. So, we have this O2 concentration on the x-axis and this partial pressure, or basically like the amount of O2 and on the y-axis. Sorry, O2 concentration on the y, partial pressure on the x. This O2, amount of O2, concentration in the blood. So, these diversities are important for different animals. So, like, basically, the physiological demands of specific animals is going to determine the pigment that they use. So, it is important to be able to evaluate and interpret these types of plots right here, these curves. So, this will come up later in a later slide. So oxygen binding hemoglobin. So just some terms to note: oxyhemoglobin is oxygen ligated hemoglobin. Deoxyhemoglobin is no oxygen bound. Methemoglobin is an iron atom oxidized to the ferric state, which cannot bind oxygen. Normally, the iron is present in the more reduced ferrous 2+ state. So, methemoglobin reductase can absorb hemoglobin to the ferrous state. Then we have carboxyhemoglobin, saturated with carbon monoxide, CO, and it says 200 times greater affinity for CO than O2, which is why you can get carbon monoxide poisoning very easily, because it has a 200 times greater affinity for the hemoglobin, and of course, promotes chemical asphyxiation. Then we have carbanohemoglobin or carbaminohemoglobin. This is CO2 bound to hemoglobin. So, don't get too caught up in the terms. They'll make more sense when you use them, and you can kind of work your way to the term once you've figured out the structure of hemoglobin a little bit better. So, oxygen binding promotes a critical conformation state in hemoglobin. This is very important to understand the binding curve and just the overall role that hemoglobin plays. So, we have the tense state when it's not saturated with O2. We have a relaxed state when it's saturated with O2. So in the tense state, only the alpha chains are available for oxygen binding. There's 8 salt bridges present that stabilize the fully deoxygenated hemoglobin in the tetramer state. As the first oxygen, then second oxygen bind, some of these non-covalent electrostatic interactions are disrupted. So after the first oxygen binds, the second one will bind easier due to the fewer number of salt bridges, right? And then when 2 oxygen molecules have bound, the full T2R conformational change occurs. Presumably at this point, sufficient numbers of salt bridges have been broken to allow full conformational change to the R state. So what does that mean? So we have these salt bridges, right? Could I find it better? No. So we have these salt bridges, right? Here on these heme groups, and basically they're stabilizing this heme group, right? And usually in chemistry, if something is stable, it's less likely to react because things prefer to be stable, low energy states. So when we have this first oxygen bind and then the second oxygen subsequently, these salt bridges are disrupted, and it's going to increase the disruption of these salt bridges, which is going to make it less stable and thus more likely to bind the oxygen molecules, right? So when 2 oxygens are bound, the full T2R conformational change, with the tense to relaxed conformational change occurs. So basically all you need to know is that deoxygenated hemoglobin has less of an affinity for oxygen than 1 or 2 oxygens bound, right? So the more oxygens that bind, the conformational change occurs to basically promote more oxygen binding. Hopefully that didn't get too confusing. So the conformational change to the R state allows oxygen accessibility to the 2 beta chains, therefore promoting the binding of the next 2 oxygens. So the third and fourth oxygen bind essentially at the same time. Triply liganded hemoglobin cannot be identified biochemically or supported by thermodynamic grounds. So basically what that means is once the first 2 oxygens have bound, this promotes such an affinity and such a rapid affinity for oxygen in the relaxed state that essentially the other 2 oxygens bind at the exact same time. So the relaxed state is immediately going to bind the other 2 oxygens, fully oxygenated hemoglobin right here. And this model for oxygen binding in human hemoglobin is based in a large measure on the benchmark research by the later Professor Stan Gill. Yeah, you can go ahead and look up this paper if you want. It can maybe clarify some things or maybe confuse you more. So whatever you think. So that is the hemoglobin binding, the chemical structure, this explanation for what comes later in these curves. So cooperativity of oxygen binding by hemoglobin. What is cooperativity? So basically when oxygen binds at one site, like I said, facilitates the binding of the subsequent oxygen molecules. This occurs in proteins with multiple ligand binding sites, usually multiple sum units. And this is usually a conformational change affects the ligand binding at the succeeding sites. So, it's very important for physiology because cooperativity changes the shape of the oxygen curve. It makes it more sigmoidal or S-shaped. As you can see right here, kind of like this S shape. The sigmoidal shape of the hemoglobin oxygen binding curve is due to the cooperativity that results of the homotrophic interactions. So, these homotrophic interactions is due to one type of ligand impacting the binding of additional molecules of the same ligand, so homotrophic. So the ligand, i.e. when these O2s are binding to these heme groups, and they're all heme groups, so the 4 heme groups, homotrophic, same ligands. So each oxygen binding to each ligand site impacts and increases the cooperativity, increases the subsequent oxygen binding to be more likely, essentially, making this S sigmoidal shape, right? So this is like, this is a good equation to know. Not exactly what I would call, I wouldn't exactly call this very, very useful in like a day-to-day, right? I mean, I guess none of this really is, but this is just kind of more to help you understand if you're a more math-oriented person. We have the Hill equation, which quantitatively describes the degree of cooperativity displayed by the pigments. So log y over -1 is log k plus n log PO2. So y is a fraction of sites oxygenated. 1 minus y is a fraction of sites not oxygenated. K is the binding constant. Partial pressure of oxygen is PO2, and N is the Hill coefficient, which measures the subunit cooperativity. That's the important thing to know, is the Hill coefficient is the subunit cooperativity. It ranges from 1 to 4 based on the slope of the plot. So, of course, here, this N is going to be lower than this because the slope is more sharp, or more, I guess what you would call like a higher degree. So basically, the S-shaped binding curve with cooperativity steepens. That's the word I was looking for. Steepens the slope across the critical range of PO2, or partial pressure of oxygen. So this makes hemoglobin very sensitive to changes in oxygen, like we described before. So another just like good slide, good picture to note: oxygen binding affinity. The partial pressure at which hemoglobin is 50% saturated, P50, as we can see, the partial pressure of oxygen, saturation of hemoglobin at 50%. The sigmoidal shape due to cooperative steepening to the slope around the P50. So we have this like, right here we're having like 1 oxygen, maybe 2 oxygens here exactly, of 2 of the 4 oxygens, 50% of the oxygen saturation, or 2 of the 4 binding sites bound. And of course, as you can see, immediately after the 2 subunits are bound, or the 2 heme groups are bound to oxygen, we're going to see a very sharp and steep increase, which would represent the last 2 groups being attached to oxygen very quickly. Again, sigmoidal shape. And the change in the hemoglobin concentration can alter the O2 capacity of the blood. So obviously, millimeters of oxygen to blood, and we have the PO2. So with high hemoglobin, we're going to have more oxygen saturation, or higher carrying capacity, rather, due to the solubility issues previously described. Now, there are also heterotrophic allosteric effectors of oxygen. Heterotrophic interactants going to be basically the opposite of homotrophic. The binding of one type of ligand impacts the binding of a different type of ligand. For example, the binding of protons to hemoglobin profoundly affects the properties of the oxygen binding. So, proton binding to hemoglobin stabilizes the tense state, fosters less oxygen binding, and therefore lowers the affinity, right? So, if you, instead of having these oxygens bind to these heme groups in this hemoglobin, if you have protons binding to these salt bridges, you're going to stabilize the tense state, and it's going to basically make it less likely to make this conformational change to the relaxed state, and less likely to bind oxygen. So what does this mean for our right shift in the binding curve, right? This curve shifts to the right, essentially making it to where more oxygen is needed to saturate these hemoglobin binding sites in the presence of reduced pH, or higher protons, because again, these protons are binding the hemoglobin and stabilizing the tense state. So it's going to take more oxygen for the hemoglobins to bind the oxygen, right? So what is the root effect? Seen in hemoglobins of many telost fish, but not in other vertebrates, some crustaceans, cephalopods. It's basically a reduction in the maximum carrying capacity of blood, right? So the max percentage of saturation of these pigments in response to lower pH. So right here, as a higher pH, there is there's a lower maximum saturation. So the curve does shift some, but it's more that you're seeing that this line, this asymptotal effect right here, when it flattens out, will just be lower with lower pH. That's the root effect. That the carrying capacity is lowered with lower pH. In the Bohr effect, the shift is curved to the right. The curve is shifted to the right with lower pH. Okay, so what is the physiological importance of this Bohr effect that we were speaking of, right? So obviously at O2, partial pressure at 29, the right shift promotes the unloading of oxygen, right? So a lower pH is going to promote the unloading of oxygen. Why is this important? Well, in lungs, when CO2 is exhaled in the lungs, we are going to have a higher pH and thus a higher binding affinity for oxygen. The curve is going to be more to the left. So this is going to promote uptake of oxygen into the blood while you're breathing in your lungs. And your tissues are going to be a little bit lower pH, higher proton count, or higher proton concentration. And this is going to promote the offloading of oxygen, or the release of oxygen into the tissues. So we're getting a higher uptake of oxygen in the lungs and a increased offloading of oxygen in the tissues where they need to be. So that's kind of just like the basic importance of why the Bohr effect exists, or rather why it's important for physiology. Okay, so this is kind of just a repeat of what I said, but we'll go ahead and read it out. The pH of the blood is lower in the metabolically active tissue like skeletal muscle, where the pH is more alkaline, the respiratory surface. Thus, blood circulating through the system, the Bohr effect promotes the unloading of oxygen at active tissues and reloading at the respiratory surface. So basically what I said about the lungs and the tissue, like muscle. In humans, 2,3-diphosphoglycerate is formed in an unusual glycolytic sequence inside the red blood cells. And low oxygen in the red blood cells causes a Pasteur effect, which increases the levels of 2,3-DPG. These elevated levels shift the oxygen binding curve of hemoglobin to the right, which we'll talk about on the next slide, thereby facilitating oxygen unloading in hypoxic regions of the body where oxygen is most needed. So these are two importances. This is more about the Bohr effect. This is a secondary mechanism by which we're going to get offloading of oxygen from the hemoglobin in hypoxic regions of the body, and that will be right here. Okay, so the 2,3-DPG effect is seen in mammal and bird erythrocytes, or red blood cells. So inositol hexaphosphate in some birds and turtles, and ATP, GTP, and ITP in various fish species serve to increase the right shift of oxygen binding curves like 2,3-DPG in mammals. Just a little bit of a side thing right here. So these organophosphates stabilize the T state, just like we had with the protons in the Bohr effect, stabilizing the T state. So if we're stabilizing the T state, we're decreasing the affinity for oxygen binding on the hemoglobin, because what promotes the oxygen binding is the disruption of these T states by oxygen binding these salt bridges. So the organophosphates stabilize the T state. Respiratory pigment 2,3-DPG binds to, i.e., in between the beta chains, 1 molecule per tetramer. In contrast, the protein binding sites are found on both beta and alpha chains. So this is just going to bind to the beta chains, but nevertheless, we're going to see that a high 2,3-DPG is going to again shift our oxygen binding curve to the right, and it's going to increase the affinity of hemoglobin. As the 2p3 increases, the O2 decreases and the P50 increases, right? So what does that mean? As the 2,3-DPG concentration increases, the affinity of hemoglobin for the oxygen is going to decrease because we're stabilizing the T state. So oxygen availability or partial pressure is going to drop across the pathway for delivery. So despite all the gas transport mechanisms we've talked about, the driving force for diffusion is reduced, and consequently mitochondria are routinely exposed to low oxygen. During high metabolic demand or environmental O2 restrictions, mitochondria become very O2 limited. So we can see that the partial pressure obviously in the ambient air is high, and as it reaches the alveoli, it's going to get lower. Arterial blood, the capillaries, and then by the time it's reaching the mitochondria, and again, note, the mitochondria have a high affinity for O2. The low O2 availability is typical for the mitochondria in vivo. So it kind of does just make sense if you look that the availability of oxygen as it's being transferred from the outside air through the alveoli, the arterial blood, and the capillaries into the mitochondria of the cell, it's going to become lower and less available, even with these transport mechanisms we've talked about above. Okay, so carbon dioxide transport. Carbon dioxide is more soluble in body fluids than oxygen. However, little free CO2 is actually transported in the plasma, only about 7% in total in humans. Some CO2 binds blood proteins, i.e., the term we talked about, carboaminohemoglobin. It's about 10 to 20%, but most CO2 is transported as bicarbonate. And so we can see this equation here that CO2 and water are going to be catalyzed into carbonic acid and bicarbonate with a proton on the end. And this is catalyzed by this enzyme carbonic anhydrase. Again, catalyzing the formation of this bicarbonate. And now we can talk about the importance of this, because this is actually an important mechanism for regulating this chloride shift, as it's known. Okay, so carbonic anhydrase is located primarily within the red blood cells. So this reaction to synthesize bicarbonate occurs in the red blood cells, even though most of the bicarbonate is actually carried in the plasma. So how is this done? So basically, this bicarbonate is exchanged with chloride across the red blood cell wall via this band 3 protein, which results in this so-called chloride shift. So we can see that this carbonic anhydrase is going to catalyze with CO2, and H2O is going to catalyze into a proton and this bicarbonate. And so this catalyzed by this carbonic anhydrase, and then we're going to have a chloride shift where this this exchange of bicarbonate with chloride is going to carry it out of the red blood cell into the plasma. So this reaction is occurring in the red blood cell. However, the majority of this bicarbonate is actually carried in the plasma. So CO2 from the outside diffuses into the red blood cell, catalyzed by carbonic anhydrase into the bicarbonate, and is exchanged via a chloride shift across this this protein, this Cl-HCO3 exchanger, into the plasma. Okay, so that brings us into the Haldane effect, which is the relationship between the CO2, the partial pressure of CO2, and the total CO2 content, including bicarbonate, of the blood. So deoxygenated blood carries more CO2 than oxygenated blood. This is basically what the Haldane effect is. So deoxygenated blood carries more CO2 than oxygenated blood. CO2 binds deoxyhemoglobin, which, if we go back up to our terms right here, just if you need a refresher, deoxyhemoglobin, no oxygen bound, right? So when there's no oxygen bound, we're going to have this increased binding of CO2 to the hemoglobin. And this reacts with the terminal amino acids of the proteins, changing the form change the forms to negatively charged carbamate groups. So this interaction fosters salt bridges that's, again, like last time, stabilizes the T state, promoting oxygen release. So if you ever see anything that is stabilizing this 10 state, you're going to notice that it's going to promote oxygen release and reduce oxygen affinity. So CO2 is another heterotrophic ligand, right? Heterotrophic, different, one binding of one ligand affecting the binding of another ligand. And in this case, it's CO2 affecting oxygen. Heterotrophic, right? Okay, so acid-base physiology. How are we buffering our blood pH, and how is this accomplished? It's accomplished by blood proteins, primarily hemoglobin, but other plasma proteins as well, like albumin. And as we've seen, the bicarbonate pool in the blood is very large and can particularly serve as a major source of buffering itself. However, the pK values of bicarbonate are not suitable for buffering the blood pH, which is around 7.4. To be an effective buffer species, the pK should be within 1 pH unit of the prevailing pH. So the pK of 6.2 is only marginal. Thus, there's little buffering effect per mole of bicarbonate, but its high concentration does produce a modest effect. So how does this work? The major importance of the HCO3 system in blood pH is that it can be rapidly changed. So when we are exhaling via when we are raising the CO2 via ventilation of the lungs, gills, it alkalinizes the pH. So we're removing CO2 from the blood, increasing the pH. We can lower the pH and make it more acidic by excreting the bicarbonate out of the body via the kidneys or etc. So just for example, kidneys, right? So removing CO2, raising pH from the blood via ventilation, and lowering the pH by excreting the bicarbonate out of the kidneys, or just excretion of bicarbonate in general is going to lower the pH, make the blood more acidic. Okay, and this brings us into this thought process that in the early days of physiology, we thought that studies showed that there was this tightly regulated pH of blood around 7.4, and this is just a special case for constant body temperatures. In ectotherms, or cold-blooded animals, the pH is maintained by a regulatory mechanism in the temperature-dependent manner. So it's not regulated at a constant value. And this does get a bit confusing, but at least to me sometimes. But let's just get into it, all right? Blood pH is highly temperature-dependent in ectotherms. So neutral water pH is defined as the pH of pure water. The neutral water pH is not always 7, but rather varies with temperature. This variation reflects H2O's tendencies to dissociate into protons, and OH as temperature is raised. So recall that H actually exists as H3O, hydronium ions in water. So just a little bit of a chemistry jargon for you there. Interestingly, blood pH is actively regulated by the animals in a way that parallels this change in the neutral water pH, but the pattern is simply shifted more alkaline as a constant relative alkalinity is maintained in the blood. So this pH temperature pattern curve is often referred to as the Ron-Reeves curve. So there's a 0.017 pH unit increase in blood per Celsius decrease in temperature. So we can see this curve here. We have our body temperature as it's getting, how do I explain this? So as this temperature, let's start over here because that's how I said it here. 0.017 pH unit increase in blood per Celsius decrease. So if we're looking from the right to the left, the Celsius decreasing from 40 to 0. And we see that this blood pH curve, right, is increasing. So as we're getting lower in temperature, this pH is shifting upwards. The blood pH is shifting upwards. And these dots are just like representing the different species, but there is just this general curve upwards. And you can see also that it parallels the neutral pH of water, pure water, going up as the temperature decreases, right? So, relationship also holds for intracellular components. So, it's not just the blood, but like this temperature change is going to just increase the overall pH of the intracellular compartments as well. Again, temperature is decreasing, pH is increasing at a rate of 0.017 pH units. Okay, so what is alpha stat regulation? Okay, so we're gonna throw out this fat jargon real quick, and then we're gonna explain what this means. Alpha stat regulation, by definition, is the adjustment of intracellular and extracellular pH in a manner that maintains constant the fractional dissociation state of histidine in proteins, like alpha-imidazole, as the temperature changes. So, as temperature falls, the pK of histidine imidazole groups in proteins increases passively, very similarly to the neutral pH of water that we talked about previously, and i.e., the pK increases about 0.017 pH per unit of 1 Celsius decreased in temperature. So, in better terms, the histidine tends to bind more protons at colder temperatures. So at lower temperatures, we're gonna have this increase in pH, or as temperature falls, we're gonna have this increase in pH, and so there's gonna be this increase in the pK histidine and imidazole groups passively, very similar to water. So In both intracellular and extracellular fluids, in previous slides, the prevailing pH was regulated upwards. That is this Ron Reeves curve we saw. By the animals as the temperature falls to offset or compensate for the inherent tendencies of imidazoles to take up protons due to the increased pK at lower temperatures. So, if unopposed or without an upward regulation of pH, proteins would bind more protons at lower temperatures, and as a consequence, the fractional dissociation state of imidazole would change, and this would have terrible effects for the cell. This change in protonation would potentially disrupt many important functions of the properties of the proteins, particularly enzymes. I found I spent a few minutes trying to type out a very condensed explanation in simple terms of what I was talking about in the alpha-stat regulation, because it's kind of hard for me to explain, without thinking about it and writing down what I think. But alpha-stat regulation is the physiological strategy allowing the blood pH to rise during the cooling. So, blood pH is rising during lower temperatures so that the ionization state of histidine residues in proteins remains constant. This helps preserve the enzyme activity, protein structure, and cellular function, etc. So, all you have to know, blood pH rises due to cooling so that the fractional dissociation state of histidines can remain constant as temperature changes. That's basically what alpha-stat regulation is, right? So, alpha-stat regulation, continuing, however, the pH is regulated upward as prescribed amount of body temperature falls. So, there basically is, again, like I said before, no change in alpha-imidazole. So, again, note that many enzymes have histidine residues, and changes in protonation states of these histidines would disrupt substrate binding. So, functional characteristics of enzymes, the Km, or the Km value's preferred reaction direction, are preserved due to alpha-stat regulation as temperature changes. So, while initially it seems that pH is being regulated in ectotherms in a way that is inconsistent with homeostasis, it's the fractional dissociation state of histidine in proteins that is being regulated by homeostatic manner as temperature changes. So, basically, what this is just saying is that it's not so much the pH that is being regulated, it's the fractional dissociation state of histidine that is the importance. This is why the pH is being regulated, because without this fractional dissociation state of histidine being kept constant, there would be some perturbing effects on these enzymes and these alpha-imidazole groups, which would disrupt enzymatic function, cellular shape, etc. So,